so long. I want to say that's the soma. I want to say that this is an axon. I want to say that's a soma. But again, I don't remember. I want to say that's a
Okay, soma, this one.
I think so. It's been so long since I took this course.
Oh, wow.
So, by like so long, like a year ago, year and a half ago. It was longer, but.
Everybody get the handout?
Yeah.
All right.
Are you taking your exam?
Hmm?
Are you taking your second exam?
No, I'll probably take it tonight. I just don't know what time. So.
All right, if you're keeping track, how many tissue types have we talked about up to this point? 3, right? We've talked about epithelium, connective tissue, and muscle. So now we're going to add our 4th tissue, nervous tissue. So let's go over some general characteristics of nervous tissue. 2 main cell types. 1, neuroglia. And 2, neurons. Okay? 2 big branches of nervous tissue. Neuroglia, of which there are 6 types that we're going to talk about. And neurons, a few different varieties of neurons. Okay. Neuroglia are support cells. Neurons are conducting cells. So the neurons create and conduct action potentials. The glial cells provide support. Used to be, we teach that you had 10 times to 100 times as many glial cells in the brain as you do neurons. We think nowadays that ratio is probably closer to 1 to 1. I think it's probably about as many neurons as you have glial cells. So, I guess, in short, we don't know for sure. Okay. You probably have a few more glial cells than you have neuron cells. The neurons are what get all the attention, because these are the ones that create and transmit the electrochemical impulses we think of as nerve impulses. The proper name for a nerve impulse is an action potential. Okay? I think of it like this. Movie stars and rock stars and stuff like that, they get all the attention. But they really aren't going to be able to carry on their big shows and stuff like that without all the support people, right? The lighting people and the costume people and the engineers and all that. They don't get any attention. You know, you sit around and do you watch the movie so you can watch the credits at the end? No. Okay. Maybe there's an end credit scene. But for the most part, those are the unsung heroes that make all that work. That's kind of the relationship between neurons and glial cells. Glial cells are the ones that are there. They have an incredibly important function, but they get ignored most of the time. Okay? The neurons are the ones that create and transmit the impulses. So why do the neurons get all the attention? Well, because our nervous system consists of 3 basic functionalities. Sensory input, so sensing our environment and sending that into our central nervous system. So when we talk about input, that's what we mean. We mean taking information from the periphery and sending it into our spinal cord and our brain. Integration of that information, comparing it to something, taking multiple impulses and making some kind of cohesive response out of it, that's integration. And that happens within the CNS, the brain and spinal cord. And then finally, what we call outgoing information or output, and we call that motor. Okay? Sensory information is coming in, we integrate within the CNS, and then we output a response, and that's called motor output. This should sound an awful lot like the parts of a negative feedback loop, where we had sensory information recorded by a receptor, transmitted via afferent pathways to the control center. The control center did integration, then communicated with the efferent pathways to the effectors. That sound familiar?
Yes.
It's exactly what's going on here on a larger scale. Sensory input coming in, integrated in the CNS, and then motor output. True or false? Motor output means movement. True or false?
False.
False. That's not what motor output means. Motor output simply means output. It means going out to an effector. There are two types of effectors, right? Muscles and glands. So only one of those produces movement. That's muscles. Glands don't. Okay? So motor output means to control an effector, okay? Or to target something. It means output. That's all motor means. And I know that goes against what you have ingrained in your brain, but you need to get that rock solid. That motor output, all motor output means is just that, output. Controlling skeletal muscles, yes, that's output, so that's motor. But controlling cardiac muscle, smooth muscle, glands, and actually activating other neurons is also motor output. Okay. So that's why neurons get all the attention, because neurons take care of that. Neurons do the sensory input, they do the integration, and they do the output. But they're not going to do any of that without all the protection and aid they get from the glial cells, so they work together. So we're going to go over neurons today in some detail. There's a neuron. And that's what the model you have on the desk is of. The cell body is called the soma. Soma is Greek for body. So the cell body, any cell body is called a soma. In the neuron, there are specific things that happen on that cell body, so we draw attention to it. Projecting off of the cell body are short extensions called dendrites. Sometimes dendrites are called receptive endings. Then there's the soma, and then usually, traditionally, there's one big long extension coming off of the soma, and that's called the axon. And the axon ends in a terminal. And sometimes that's referred to as a bouton. Or little booty. Okay? So that's the axon terminal. With me on that? So what do we have in the soma? Well, the soma is going to have all the traditional organelles, so it's going to have a nucleus in there. And the nucleus is going to be studded with nucleal pores. And there will be that denser region in there known as the nucleolus. nucleus, nucleopores, nucleolus, and you can see that on your model. If you look in there, you'll see you've got the nucleus studded with little nucleopores. It even shows you the double membrane called the nuclear envelope. I didn't write that in the diagram.
The one
And
in
then
the
that
middle
denser
is
region in there of the nucleolus.
Easy here.
Yeah.
Maybe.
Red
Oh.
thing. I think a rough ER. I think so. Yeah.
You have extensive rough endoplasmic reticulum inside neurons. There's an eponym for that, it's called Nissl substance. So when you look inside there, you see this gray material with all the little red dots on it. Okay? That's called Nissl substance. It's so thick and it's so elaborate that you actually can see it in a light microscope, which is pretty unusual. You can't usually see rough ER in a light microscope. So these things called Nissl bodies or Nissl substances, the remnant, not the remnant, but the observable part of the rough ER. Scattered around in there, you have these very long mitochondria. They look a little bit like red worms. We have plentiful mitochondria. So right away, what does that tell you about a neuron? It has a tremendous amount of rough ER and it has a lot of mitochondria. What can you start to make some predictions about its physiology then? You think neurons are going to do a lot of protein synthesis? Yeah, because that's what's made on the rough ER. Okay. So if you have a lot of rough ER, then it probably means neurons do a lot of protein synthesis. The fact that the ribosomes are on the ER means that those are probably secreted proteins, because that's what you manufacture on the ER. And do you think neurons have a high energy requirement?
Yes.
Yeah, if they have a lot of mitochondria, then yes, they certainly do. And those are two basic principles of neuron physiology. Okay. I asked you to read about the cytoskeleton components and most other organelles, because I didn't really go over that verbatim in lecture on each one of those. The cytoskeleton components consisted of the microfilaments and the intermediate filaments were actin, and they were structural components of the cell. But then there was this larger cytoskeleton component called a microtubule. And microtubules are made out of alpha and beta tubulin proteins. They're much larger. They're a good 10 times larger in diameter than the actin filaments, and they're hollow. And these microtubules are mostly used for intracellular transport. They're like railroad tracks inside a cell. And the little railroad cars that you load onto them are vesicles. So you already know something about what vesicles are, little lipid envelopes we use like Tupperware containers. So, if you're following the logic here, we manufacture a lot of secreted proteins here on the ER. They're probably going to get processed in the Golgi body, which I'm not going to draw on there. And then they're loaded onto these microtubules for transport. Well, where are they going to go? Good question. So these microtubules you can actually see streaking through the cytoplasm, and so they get the name neurofilament. And this kind of run the length of the axon. I'll draw a little vesicle in green here. So maybe we make some vesicles. And those vesicles get loaded onto these microtubules. And they make their way down here. to the terminal. And these vesicles, among other things, will probably contain neurotransmitters. So we manufacture this stuff up in the soma, but we're going to release it down at the axon terminal, which means it has to get there somehow. We could depend on just diffusion, but that would be horribly inefficient. That would be like manufacturing something at a manufacturing plant in Cleveland and expecting it to build up in big piles, and eventually that pile reach all the way to Columbus. You know, while in theory it's possible, that would not be a good mechanism for doing it. Instead, we're going to load it on a truck. You know, back when the world worked, you could load things on a truck and it would get there. So that's what we're doing here. We're loading this stuff into a vesicle, putting it on a railroad track, basically, and shipping it down here. Of course, there are names for all this. It's called vesicular transport. It's called axonal transport. If it's going towards the axon terminal, it's called anterograde transport. And sometimes we recycle the material by taking it back up here to remanufacture it, and that's called retrograde transport. So there are names for all this, but it's all vesicular transport. So we manufacture the stuff up in the soma, transport it down to the terminal. And eventually we're going to trigger the release of this material at the terminal. Okay. So dendrite, soma, axon. You good with that? The typical organelles that are in there, we emphasize some of the important ones for neuron function. Now I want to talk about one specific part where the soma meets the axon, because there's something very interesting that goes on here. So let's recreate our drawing here a little bit. Flat, right? Now I want to look at just this area right here. What I've highlighted there is an area called the hillock. So that whole thing is the hillock, and I put TZ and IS. Okay. TZ stands for trigger zone, IS stands for initial segment. Parts of the hillock are the trigger zone and the initial segment. And they're right at the junction between the soma and the axon. So go ahead and put your finger right on the hillock on your model so I know that you know what I'm talking about. Okay. Right. Let's see.
I wouldn't say it's like this area.
So the part that's closer to the soma, where the soma tapers to the axon, that's the trigger zone. Okay, so that's going to be more
Trigger zone and then
this
initial
is
segment. This is the soma.
I mean, see, there's no actual line between the trigger zone and the initial segment. It's a functional difference. But once you have that tail that's kind of sticking off of the cell, you're on the axon at that point. Okay, that's the initial segment. And the part that's still on the soma and kind of tapering, that's the trigger zone. So if we're going to, so this is the trigger zone here, and this will be the initial segment. Right?
Trigger zone, initial segment. Correct. Trigger zone, initial segment.
Yep. All right. The reason we're focusing on that detail is because what's going to happen in that trigger zone is going to determine the overall function of this neuron. It's the most important functional part of the soma. Okay?
Trigger, initial
So you got the
segment.
architecture down. That's what we're going for right now. So what's the job of a neuron? little bit louder.
Output something.
Between the air handlers up here and the mask.
To create an output, right?
Create an output, okay. What'd you say? Make what? What makes its way down there? The arrow? Oh, well, so the whole thing's the neuron, right? Dendrite, down to the axon. Something does make its way down to the terminal.
The vesicles or neurotransmitters.
Ultimately, we're going to trigger the release of neurotransmitter.
Oh, action potential.
Action potentials, there you go. So way back at the beginning when I said neurons generate and transmit action potential, remember that? Okay. So we're starting to forget the trees for the forest barrel. Okay. We're looking at all the details, but we're forgetting the point. Okay. The point is create and transmit action potentials. Now everything you guys said is true. Okay. We're eventually going to trigger the release of neurotransmitter down here from the terminal. So something's going to make its way down to the terminal. That's absolutely right. That something is an action potential. All right, so we're just going to draw one terminal on there. There's our axon. So we're going to get some stimulation at these dendrites. That's why they're called receptive ends. Those kind of golden arrows represent what we call local potentials. I'm going to leave it at that for now. I'm not going to define what those are because that's going to take the rest of the class to get into that. But a potential is a measure of a voltage. So this is an electrical stimulus of types. I tried to indicate it's kind of big here. I didn't do the best job because I didn't want to change pen sizes. And then it gets smaller as it goes across there. We call that being transmitted in a decremental fashion. Okay? Think about it like sound. Okay? If you're right next to me, I'm shouting. If you're 20 feet away, I don't sound so loud. It's getting softer the farther you get away from the initial point, right? That's what we mean by in a decremental fashion. So these disturbances or stimuli are large here. By the time they get transmitted over there, they're a little bit less. The job of the trigger zone is to do summation. Summation. Okay? A neuron's response when it comes to the axon is all or none. Give me an example of an all or none response.
Turning on a light switch.
Turning on a light switch. Mostly, but what about a dimmer switch?
Okay.
Okay, so yes, on, off, right, but no dimmer switch here.
Starting
so that
a
could be an example.
Starting a car?
Yeah, okay, the car's either running or it's not, sure, okay. The old Pink Panther, not old, but the Pink Panther movies with Steve Martin, you ever see these? There's a great line in there where one of the guys gets killed and Tauntaun says, oh, it was fatal, and Clouseau goes, how fatal? Completely, right? Fatal kind of means complete, right? You're dead. Funny moment. Anyway, that's an all or none. Either you're this or you're that. Okay? You're either alive or you're dead. There really is no in between. Yeah, you can be dying, but at some point you're alive and then you're not alive anymore. All or none responses, no gray area. Okay? Axons function that way. It's all or none. It's all or none. Now, given the fatal idea there, can you be dying?
Yes.
Yeah, you certainly can be. You can be dying for a long time, right? But ultimately, you reach that point where either you're alive or you're dead. So these stimuli that are coming across here, I don't know why I picked this morbid example, but that's kind of like dying slowly, okay? The trigger zone decides well, are you finally dead or are you alive? Okay? And then once it makes that decision, it relays that message down the axon. It could be like, the way I always try to think of it is, I have 4 kids, and they're all older now, but back when I was younger, you'd host a birthday party or something like that, and there's nothing more fun to do on your Saturday afternoon than have like 12 screaming middle schoolers, maybe, you know. Headaches. So you're driving along, you know, and you've got all these kids back there in the back, and they're all making noise and whatnot, and you as the driver, you know, you're trying to pay attention. I remember coming home from a birthday party once. We had a horrible snowstorm. You couldn't see the roads. Everything was horrible. You know, of course, the kids don't care. They're in the back making noise, and you're driving. You're like, oh, I just want to get this over with. And at some point, somebody back there makes so much noise or does something so obnoxious that you just snap. You know, you're like, you turn around like, shut up! And you just try to smack whatever you can reach. Up to that point, you've been maintaining your cool, okay? But then finally, you broke. Now, these are all the kids out here. These are all the stimuli. And you've got 4 or 5 up back there that are just making obnoxious sounds. And you've got the one good kid back there, which I'd like to say was mine. He probably isn't. You know, and he's back there going, shh, because this is like the one kid that's aware that, you know, there's bad weather and we could all die. And so they're in the back going, like, shh, calm down, shh. Right, can you picture this? Right, you've got the 4 or 5 that are being rowdy, and then you've got the one that's always like being like, come on, calm down. So these stimuli that come across here, there's no rule that says these stimuli have to be excitatory. Some of these stimuli are inhibitory, and we'll talk about how that works later. But they're all being transmitted to this trigger zone. The trigger zone, you're the driver, you're in control. At some point, you're going to respond, okay? Maybe you make it all the way to your destination and you never flip out. Great, you didn't respond. But you're driving along and then something finally tips you over. It wasn't just 1 kid or 2 kids or 3 kids. It was all of them. And it wasn't just one time, right? We've all had that unpleasant experience in our life where we've lost our cool, and it wasn't the one thing that was said right at the time, was it?
No.
It was the hours and minutes and days that led up to that. Okay? And it was funny, that one little trigger that's like, I just can't take it anymore. That's what we're talking about. Okay, these types of stimuli are all adding up here. So when I say summation, that's what I mean. Of course, we're not doing it with words and noises and stuff like that. We're doing it with chemicals, but the end result is exactly the same. There is something called a threshold. And when you hit that threshold, the axon responds. Okay? And that's it. It's all or none. Either you get to that threshold or you don't. Okay? In the example I'm talking about, you know, flipping out while you're driving your car, do you always have the same threshold? Do you always have the same threshold for you to respond?
No.
No, right? If you haven't had enough sleep and you're over-caffeinated, your threshold is way down here. If on the other hand you had a really good day, you got a bonus in your paycheck, and you got a good night's sleep, well, your threshold's probably up here. Okay? You know? We're somewhere in between, depending on how life is treating us. You think that kind of thing happens to these too? Yes. Threshold will move a little bit. So that's why neurons don't always respond the same. Are you with me as far as what we're trying to convey? I'm trying to stay away from all the nuts and bolts right now. I'm just trying to give you what it does. So when we say integration, that's more than just a word. Okay? This is what we mean. We mean all of these local potentials are being transmitted across the soma to the trigger zone, and the trigger zone has a special type of receptor in there, which it has thousands of, that makes it capable of responding and doing this summation. And then once it decides, once it hits threshold, then boom, it relays the signal. Okay? So the trigger zone creates the ultimate signal, which is known as an action potential. So it got created there. That's where it arose. And then you notice it bunny hopping down the axon. So axons transmit action potentials. According to my drawing, do dendrites transmit action potentials?
No.
No. Does the soma transmit action potentials?
No.
No. Only the axon transmits action potentials. Okay? Big difference between local potentials and action potentials. Local potentials are decremental, which means they get less and less and less intense the farther you go away from their initial point. And they can be transmitted on dendrites and so on. Action potentials are always of the same intensity, and they're only transmitted on axons. So each one of my little bunny hops, I didn't draw it perfectly, but each one of those little bunny hops is supposed to be the same height.
Are local potentials the same thing as like graded potentials?
Graded potential?
Or is it the same thing or
Yeah,
not?
yeah. Graded and local potentials. Another word for local potential is a graded potential. G-R-A-D-E-D. Okay. So the trigger zone is actually part of the soma, where the action potential gets created. The initial segment is where it first gets transmitted. So the initial segment is part of the axon. The trigger zone is the end of the soma. Okay. The two together are known as the hillock. Well, there are different ways to transmit action potentials down axons. We can do it continuously, or we can do it discontinuously. And one of these days I'm going to remember to bring in some dominoes so I can demonstrate this properly. In today's digital age, do you guys play with dominoes anymore when you're kids?
We
Building
have
blocks,
a
dominoes, right?
box of dominoes.
Okay. You know, I'm sure there are kids out there, it's like grandma and grandpa have dusted off the dominoes and they look at them and go, what's that? Okay. So dominoes are these little brick-like things, okay? You stand them up, and of course you can actually play a game with dominoes, but most people don't do that. You make a little train, a domino train. So there's one here, there's one there, there's one there, there's one there, there's one there. Bear with me, there's a reason I'm talking about this. Domino 1. Domino 2. And so on and so forth. So we can put a whole bunch of these in a row. We're just going to show them from the side. So here I've got a finger and there I've got a bell. And I want to transmit a signal. If I tip over this domino by pushing it with my finger, what's it going to do?
Knock over the rest.
Knock over the rest. It's going to fall over, right? Boom, it's going to hit this one. And then that one's going to fall over. And 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 finally, this one's going to fall over, hit this bell, and it's going to ring. Yeah?
Yes.
Fairly predictable. Did this domino ever come down here and hit that bell?
No.
That bell? No, it didn't. But knocking over the first domino did cause the bell to ring, didn't it? So when we say a signal is propagated, this is what we mean. Okay? This is called signal transduction. At no time did the first domino actually translocate over and hit the bell. That never happened. But the stimulus that was applied here did ultimately wind up causing an event to happen there, and the signal was propagated, right? Well, does that look an awful lot like the bunny hopping I put up on the axon?
Yes.
Yeah, it's the same basic idea. Okay. So, is there a way I could speed this up?
Yes.
No?
Yes.
I could slow it down or I could speed it up. I could certainly slow it down by putting in a little block. Okay. If I were to put in a little wedge in here like this, that's going to stop that. I don't know if you've ever seen this on TV. They do, you know, like they have a whole gymnasium set up like this and they knock it over. It's really cool. You should YouTube some of these videos. When the people are setting these things up, they put in these little wedges like this every once in a while. So just in case somebody happens to kick it, they don't knock over the whole thing and have to start a month's worth of setup again. So there's a way to block it by doing that. There's also a way to speed it up. Can you come up with a way I could speed this up? There's no right or wrong answer here. I just want to see how creative you can be. What if I wanted to... I'm still going to use the domino, so I'm not going to change that. But how might I speed this up?
Have less dominoes.
Have fewer dominoes?
So make it closer to the bell.
Well, okay. So if I started taking out every third domino, flop, 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 would stop. Because that domino wouldn't reach that domino. It's not big enough.
Well, like, instead of having you have 6 up there, just have like 3 up there, but like make them closer together so they can still hit the bell.
Okay, and she's being brave because she's trying to work with me here and try to answer these, so you guys can help her out. Okay, so if I take out 3 of them, put them closer together, that's going to shorten the train, right?
Yeah. Why does it have
Is it
to be
going to get
that
the signal to
long?
go from here to there if I do that?
Can we just add more force to it?
Add more force? So if I knock this one over,
I don't know.
that's a good idea. I don't think that would work.
Okay. I know what you're trying to get. You're trying to talk about like swan cells. So you need like something. You need like a bridge or a pathway to like help propel the other like force over. Or
She's
something.
pretty smart. She thinks she knows where I'm going with this, but she will. Yeah, but I'm not going to shortcut it. You're going to have to figure out the answer.
Greater stimulus?
Increase the stimulus. So that's like what he said with more force. Well, actually, no.
Make
'Cause once
something
we
to
hit threshold,
conduct.
this domino falls over, and it doesn't matter how hard you hit it.
Make something
Once you get to
conduct
enough
the
force to knock it over,
stimulus.
it's knocked over. What's that?
Make something like propeller conduct the stimulus onward after it's like terminated. Or I get what you're trying to
Look,
do.
if you were doing this, and I wasn't asking you a question about a neuron, and I said, hey, I want you to do the same thing, but I want you to get that bell knocked over quicker.
But we still have to use the
You
dominoes.
still have to use the dominoes. But maybe you can use other things. I'm not
Well,
making
that's
any
why I said
rules.
make a bridge, like on top of the dominoes, and then
Make a bridge on top of the dominoes.
and you
So
can
I
even
could
use
connect
other
this domino
dominoes.
to this domino? Something like that?
Sure, yes.
So now, if I knock this domino over, and this domino falls over, oh, look at that, it pushes this. And then that domino falls. We agree that's what would happen?
Not sure.
Sure
Sort of.
would. Okay? And that'd be a hell of a lot faster than having each individual domino fall. Okay? So, why not just have that wire go from here all the way to there? Well, because at some point it's going to get too heavy and you're not going to be able to knock it over. The stimulus won't be sufficient to get it knocked over. Okay? Now we're only dealing with 6 dominoes, so it's probably not going to happen. But if we had hundreds of thousands, that's going to become a problem after a while. It's not going to work. So you're going to be limited on practicality of how long you can make that jump. Okay. So yes, you're right. This was to talk about Schwann cells. Well, these are like continuous transmission. Domino, 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 domino. And by the way, each domino in this chain is an action potential. Every time we create an action potential, it creates a disturbance that then causes the next part of the membrane to also create an action potential. So domino, 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 domino. Well, we can put something in here which covers some of these. And that's called a Schwann cell.
Mm-hmm.
Or its newer name is the neurolemocyte. Schwann cell is the older eponym. And when you do that, it insulates the intervening region. Wait, make this small. There we go. So these don't happen.
You said it insulates
It
what?
insulates the intervening region. Now there are gaps in the Schwann cell sheath, and those are called nodes. So you look on these axons and you can kind of see that buttercream off-white wrapping around the axon. Okay, everybody look at that and identify that. Okay. The orange thing going down the middle, that orange pipe, that's your axon. So up close to the trigger zone, it looks like a cigarette. Then you go down farther, it's been cut open, and you're looking into the pink middle. That's the axon. Okay?
What did you say the Schwann cells insulate something?
It insulates the axon. I haven't discussed what it does more than that yet.
Okay, so the gap thing, you're talking about that?
Yeah, so
Okay.
that's the node. Okay? So this is the Schwann cell wrapping. That's the axon.
Which one is this one?
And then here we're getting
So this is the swan cell, The
So
node
the pink, that's
is
the
that
axon.
little thing.
See, that's the axoplasm.
So this is the node,
Yeah,
right?
that's
the
the
node.
node.
That's
So
the node.
swan cell. So this
So
is
we're
the
clear on where the
belly.
axon is, where the
Swan cell. And then this is
where the
its
node
nucleus.
This is Schwann cell.
And then the
That's the
node
nucleus of the Schwann cell.
is
That whole
that little,
white
like-
wrapping is all, this is all Schwann cell. Okay, and then that's the axon. Here, that's the axon.
Swan shell. Swan cell. The node is right here.
Very good question. Okay, so the Schwann cell is a different cell. It's a glial cell. The Schwann cell is the first type of glial cell we're going to talk about. It's not a neuron. So when you look on here, you'll see you got a little nucleus out here like this. And then you'll see multiple layers, right? You'll see it's not black like that, but it's kind of white. And that's what your Schwann cell looks like.
Nucleus for this one, so.
Okay? Schwann cell grows like a piece of electrical tape. It grows out and it wraps itself around the axon again and again and again. Schwann cells have a membrane that's made out of something called myelin. Okay? Myelin. So that wrapping that you see on there, that's called the myelin sheath. So is the myelin sheath the same thing as a Schwann cell? Not quite, okay? It's like saying a barn is the same thing as the siding on the barn. No, okay, the barn is its own thing, but mostly what you see is the red siding. So the cell grows around the axon, and its membranes are made out of myelin, so that makes up the myelin sheath. So like my domino example down below, the purpose of the myelin sheath is to speed up nerve conduction. Myelin is a fat. Fat is a connective tissue. Fat is not excitable, right? It doesn't conduct action potentials. So you're going to read myelin is an insulator. Anybody ever work with electrical circuits? Is there ever a time where an insulator speeds up electrical conduction? I was very confused when I first learned this because I was like, okay, insulators are things that block electrical flow. How the hell does an insulator speed up electrical conduction? That doesn't make a lot of sense, does it? Well, this is not truly electrical conduction. This is electrochemical conduction. So it works on a diffusion principle. All right. So now I have to get a little chemistry on you. I apologize. Sodium. Sodium ion. It's not that big. Okay, a little tiny little dot. Sodium's a monovalent cation, which means it is a +1 or positively charged ion. We're going to have more sodium outside the cell than we have inside, thanks to the sodium-potassium ATPase, something we talked about in lecture a little bit. Okay. We pump that sodium out here. Now, when an action potential arrives, it lets sodium into the cell. We'll talk about this in lecture. We'll go over how this works in lecture. So when an action potential is created, sodium comes into the cell. And that's actually what creates the depolarization phase of an action potential. Now, sodium's an ion. So we had sodium come in through a channel protein. Now, what's that sodium going to do once it comes inside the cell? Not just one sodium, but let's say there's 1,000 sodium that came in. Sodium just going to stay right there?
No.
What do you think it's going to do?
It depends on the gradient inside of what it's going inside of, correct?
It's going to create a gradient, I heard that.
Well, it depends on what it's going to do once it goes inside, because it depends on the gradient inside, correct?
Well, it's going to create a gradient. All right. We had sodium that wasn't in here, now it came in.
Right.
So now you have thousands of sodium here where it wasn't before. When you dissolve something in a fluid or in air and you put it there, does it just stay there?
No.
No, what's it do? You put food coloring into a
It dissolves.
cup. Okay, dissolve is part of it. If I take a blob of red food coloring and I put it into
Well,
a
it
bowl,
disperses. That's
oh,
the
it
word.
disperses. If only there were a word in biology we learned about that. What is it called?
Diffusion.
Oh, let's make it multiple choice. Magic or diffusion?
Diffusion.
All right, diffusion. There you go. Good job. Okay, so it's going to diffuse. So sodium's going to come in, and it's going to start diffusing in all the different directions. Okay. It's basically got 4 choices. It can go that way. It can go that way. It can magically go back out the way it came in, or it can stay exactly where we put it. All right? Of those 4 things, it's not going to do 2 of them. It's not going to go back out the channel because it can't. It's not going to stay where it came in because diffusion gradients are not going to let it do that. So it's either going to go backwards or forwards. So as it marches over here by diffusion, it's going to stimulate the next part of the membrane to open up. And more sodium will come in there. Okay? I'm not talking about how it's doing that yet, okay? But as the sodium diffuses down the membrane, it stimulates the next part of the membrane to then open up its sodium ion channels. More sodium comes in there, and we repeat the process. Where we have the Schwann cell, it prevents this from happening. So where that Schwann cell sits, it won't let any more sodium come in. It forces that ion current, that diffusion of the ion, to go all the way down to the next node, where we get another action potential. And then it diffuses to the next node. And that just keeps happening again and again and again. So that's why insulating between the nodes speeds up the process, because that diffusion is like that stick on the dominoes. The only reason this works is because this process, the ion current, is faster than continuous action potentials. Once you get to a certain distance, about 150 micrometers, it's no longer faster, and we don't do it anymore. Okay? You can do both. You can have unmyelinated axons, and they'll transmit action potentials just fine. Okay? Domino, 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 domino. Of course, they're not dominoes, they're action potentials. That's called continuous transmission, and it's slow. Discontinuous or saltatory transmission works like this, and it's fast. Okay? What do I mean by fast and slow? Okay? I'm 1.86 meters tall, okay? Give or take. Heavily myelinated axons like these can transmit at speeds of about 150 to 200 meters per second. Okay? So more than 100 times my body length in a second. That's what a heavily myelinated axon can do. An unmyelinated axon, that speed drops down to about 1 meter per second, so 100-fold slower. Okay. If you're making $1 an hour, would you like to make 100 times that?
Hell yes.
Yeah. I'd be okay with making 100 times my salary. Sure, no problem. Are there times where going slower is better?
Yes.
Well, I guess if somebody walked in the room and said, everybody that has $100 in their pocket, I'm going to beat severely, you'd be like, well, I'm glad I only made $1 this hour. You know?
Isn't it better for like more precise, like delicate actions though? You don't want to like speed that process
Yeah, I
up.
mean, you know, may God smite me with a fortune, right? But let's say you had to do accounting. It's certainly a lot easier to keep track of $10 an hour than it is $10,000 an hour, right? You got a lot more accounting to do. Okay? So sometimes speed is great, and I think when we're earning money, most of us would opt on this speed idea. But there are times where things can come too fast, right? You know, it's your job to do inventory or something like that. Right? Who doesn't have the job where somebody's trying to give them more work than they can do? And you're just like, I can't keep up. Sometimes it's all about speed. So when I'm trying to say maybe get a muscle in my finger to respond as quickly as possible, speed is of the essence. Okay? If I want to...
Isn't that like the withdrawal? Like, okay.
Well, you talk about the withdrawal reflex.
Yeah.
Yes, that does involve these types of neurons.
Okay.
On the other hand, if I'm trying to be careful and do all the proper accounting, which really for neurons is integration, then maybe I don't need that myelination. Okay? the 3 trillion neurons that make up your brain aren't too worried about speed. They're much more worried about integration. So they're not myelinated. Have you heard of gray matter and white matter? Have you heard those terms? Gray matter and white matter? Well, the myelin sheath, if you look at that, it kind of is a dirty white, isn't it? That's white matter. Okay? When you talk about white matter in the body, it's heavily myelinated. And it's all about speed. Gray matter is not myelinated. And it's all about interconnectedness. So what do you think your brain is mostly? Your brain is a mixture, but what do you think it is mostly? No. Gray. Gray, because it's the calculating part. And it wants to do all the integration. So the stuff on the outside of your brain is all gray matter. Now, your brain communicates with other regions, and when it does that, it's white matter. But when it wants to do the calculations, the processing units, that's all gray matter. But nerves, to conduct impulses from our sensory receptors in and to control our motor output, are almost all white. Questions? All right. So remember, we're trying to couch this functional stuff with the model you've got in front of you, so you have some idea of what's going on there. This model is always going to show up on your lab practical. Again, not on the one on Monday, right? Not on the one on Monday, but your next one. And I'm going to ask you just label some parts. Make sure you can tell the difference between the Schwann cell and the axon. Let's come on down the axon now. So we'll go past the nodes, the myelin sheath, and get down to the end. Your model doesn't have this on. Okay? It's cut off. Your model is cut off pretty much right there. It ends in the middle of a Schwann cell. Okay? But ordinarily, we come all the way down to a terminal, an axon terminal. That's as far as it'll go. So let's look at that terminal. I've just taken this and turned it up on end, okay? And we know in there we have these little vesicles. Vesicles with neurotransmitter. NT means neurotransmitter. Neurotransmitters can be chemicals like, and you can write this down if you want, otherwise I'm just giving you some examples. Epinephrine, norepinephrine, acetylcholine, serotonin, glutamate, substance P. There's 300 some known neurotransmitters, okay? So some substance in there. All right, what's my bunny hop supposed to be? What's that
Action
supposed to be?
potential.
Hmm?
Action potential.
Action potential, right, so that's an action potential. So we're going to say action potential is arriving at the synapse. The action potential arrives at the synapse that causes a trigger inside the synapse to cause exocytosis of neurotransmitter. What I'm denoting here is a synapse. A synapse is where two cells meet. The neuromuscular junction is a synapse. So, action potential arrived at a terminal. That's a trigger that causes exocytosis of neurotransmitter. Neurotransmitter is dumped into a gap, about a 50 nanometer gap that exists between the presynaptic cell and the postsynaptic cell. That neurotransmitter is dumped out there. Again, what does this chemical do? Just like the sodium coming inside the cell, it diffuses. Okay? In some of your chemistry training, at some point, you did an experiment with diffusion. You took a couple of molecules. I actually just walked into the human bio lab, or actually the 1040 lab, and they were doing the experiment. They were doing it with 2 different dyes of different molecular weights. And one was blue, one was red. Okay? And you put it in these little holes, and you timed it. What can you say about diffusion over time as distance increases? Faster or slower? Slower. The point of all those labs is to drive home that one point. Okay, well, 2 things. 1, heavier things diffuse slower, so higher molecular weight diffuses slower. And the rate of diffusion gets much, much, much slower the farther away you go from the original point. So you put the material in here, it diffuses from here to here almost instantly. And then from here to here, maybe never. Okay? So that distance can be a matter of a couple of centimeters. To go from here to a millimeter away, it might happen in a few milliseconds. But to go from there to 2 centimeters away might take years, or maybe never. My point is, diffusion over small distances, which 50 nanometers is a pretty darn small distance, okay, is more or less instantaneous. So the material's here, and then boom, it's there. Okay? It diffused across that cleft. It isn't truly instantaneous, right? It can be measured in fractions of a millisecond, but it goes across pretty fast. Now, if I were to expand that by just tenfold, make that gap larger, the time for diffusion would probably be measured in seconds, okay, rather than fractions of a millisecond. So we get a burst of neurotransmitter here, and what does it bind to on the postsynaptic membrane? What did I cartoon there? Oh, come on, it looks like this. What about every single time I've drawn this on a memory diagram? It looks like that, and something comes and binds to it. What is this? Time for another multiple choice question? Okay, all right. A pogo stick or a receptor?
Receptor.
See? Now this is the level, if I made the exams at this level, straight A's, right? Okay, straight A's. There's a reason I don't make the exams like that. I can tell you the names of some instructors that do. Take them. All right, so we've got a receptor over there. A receptor is anything that can bind to a ligand. So is the neurotransmitter being a ligand? Yes, the neurotransmitter is a ligand. In order for that target cell, whatever this is down here, muscle cell, gland, another neuron, whatever, it has to have a receptor for that neurotransmitter. So if the neurotransmitter is acetylcholine, then there probably had better be an acetylcholine receptor. Want to draw a little rectangle like that in a membrane, what does it always mean? Okay, multiple choice. Beer can or channel protein?
Channel protein.
Channel protein, okay. So, I bind a ligand to a receptor. Now, there's a reason that receptor is connected to that channel, because probably that channel will open or close in response to whatever that receptor is doing. Simplest case scenario, it opens. Ligand binds the receptor, channel opens. That changes the permeability characteristic of the membrane. Sodium comes in, probably can create a depolarization, and voila, we have transmitted an action potential from one cell to another. Synaptic transmission. Okay, we're going to go over this 4 or 5 more times. This is the first time I'm introducing this, and I'm introducing it in a very basic way. So if there are a lot of question marks going off in your head, that would be normal. Okay? The point of sending that action potential down the axon is to get to the terminal so we can either induce or inhibit the release of neurotransmitter from the terminal. Okay? That neurotransmitter will go across the synapse and stimulate the next cell, that effector. Go ahead.
If the next cell was a neuron, so could that act as like a stimulus to have another action potential?
Yeah. Yeah. So he asked, could that target cell, this postsynaptic cell down here, be another neuron? In which case, this would be a dendrite. And absolutely that happens. In the gray matter of your brain, in what are called association or interneurons, that's what happens all the time. Okay? And that finally leads us to one of the last things on this model we need to point out. If you look on the dendrites on your model, all right, so I said something I want you to focus on. If you look at the dendrites on your model, Is there something, is there a detail that's missing in my drawing?
Yes.
Yeah, what?
Those those things.
Those things. Those things that look
gonna
like little kind of orangish Hershey's Kisses. Those are called telodendria.
These. So this is a dendrite. These are telodendrites.
Exactly. Okay, so it kind of looks like this.
Telodendrites, dendrites.
Those things. Am I doing a passable job drawing them?
Yes.
Okay. Those are telodendria. So we probably need a really long, drawn-out description of what a telodendria is. Well, I don't have time for that, so I guess I'll make it simple. Those are axon terminals. Those are just axon terminals. All right, but Shamir, to get to your point, those are axon terminals of other neurons. Which means that this axon comes down here, that's its terminal. If this terminal were sitting on the dendrite of another neuron, then that would be this. That make sense?
Yes.
So those actually aren't part of that neuron, they're part of another neuron. Last thing to tell you. Look down at the end of the axon on your model. See that gray stuff on the very end? Okay, so two things I need to tell you about that. One, what it's called. It's called endoneurium. It's called endoneurium. And it's a type of loose connective tissue. So it's this kind of stuff that's wrapped around there. That's called endoneurium. And it's collagen and CT. And you know the connective tissues are not excitable, so it's another type of insulation. It's around the outside of the Schwann cell. And it basically is the loose packing that protects axons. So if you watched the lecture where we talked about muscle fibers, and we said that each individual muscle fiber has a little wrapping of connective tissue around it called endomysium, this is the same stuff, except it's around neurons. So it's called endoneurium instead of endomysium. It's just a loose protection. The second thing I need to tell you, and please make note of this, it's wrong on the key for this model. When you look at the key for this model, it's going to call that, I think it calls it epineurium, okay? It's not. That's endoneurium. So there's a mistake on the key. So I know students like to get the key out and they read it. And I want you to be aware that there's a mistake on that key. So that's definitely endoneurium.
But endoneurium is on the sheet though.
No, he meant like the ones that are
on this,
laminated.
but there's a printed key that came with those models, and on that key, it's labeled wrong. So if you get that key out at some point, I want you to be aware of that. Okay, I think we've gone over everything that is on the model. The plasma membrane of the axon is called the axolemma. The cytoplasm inside the axon is called the axoplasm. I think other than that, it should be pretty
How come
straightforward.
when our models they cut it off at the terminal?
Why'd they do that?
Yeah.
I don't know. I've asked myself that. The most immediate answer is because on this scale, that neuron would be longer than this building. Okay, so if they wanted to show it to you in its proper proportions, it wouldn't fit. So we have an older one, which I don't think they put out, where they have the neuron, they have the axon cut, and then they show you a terminal at the end, which I think is a better way to do it. But the other thing is, I guess it's cheaper. That's why. It'd be nice if they'd show that.
So the
I'm gonna stop there.
axolemma is what again?
The axolemma is just the plasma membrane of the axon.
Okay.
So we're going to add some histology to this next week, where we're going to look at some slides, and that's some of what else is on the sheet. And you're going to get two big neurology packets. You're going to get a packet for the brain and a packet for the peripheral nerves. And that's going to be what we do all of next week. So 3 days next week.